bacteria has been isolated as the producers of the renin. So different species, Alkaligens, Bacillus, Corynebacterium, Lactobacillus, Pseudomonas, Serratia, Streptococcus are some of the bacteria that have been exploited potential for the renin production. So there are also different fungi species like Aspergillus, Candida, Coriolus, Indothea, Entomorphophthora, Irepex, Mucor, Penicillin, Rhizopus, Sclerosium, Turulopsis are also reported to produce renin, right? So different fungi and bacterial species also they can produce these renin. Actually, production यो चाहिँ जीन ट्रान्सफर गरेर चाहिँ भएको हुँदैन है त यो चाहिँ इन्डिपेन्डेन्टली ओरिजिन भएको हुन्छ यो एन्जाइम्सहरु डोन्ट थिंक दैट यो जीन चाहिँ कतैबाट होरिजोन्टल जीन ट्रान्सफर भएर भएको होइन सो नेभर दैट कैन नेभर ह्यापन एक्चुअली के हुन्छ भन्दा इन्डिपेन्डेन्टली काल्फहरुले पनि जेनेरेट गर्ने हो इभोल्युसनरी हिस्ट्री हेर्दाखेरि काल्फहरुले सो डेफिनेटली डिफरेन्सेसहरु हुन्छ है त So different bacteria, fungi or species or broche we can produce renin. Mucor Pusillus var lind variety भनेको यो so Mucor Pusillus is this binomial system of nomenclature. यतिले मात्र अब organismsहरुलाई चाहिँ it is very difficult to recognize them and this चाहिँ यो भेराइटीहरु स्ट्रेन नम्बर्सहरु पनि जेनेरेट गर्न सकिन्छ सो यो पार्ट भनेको चाहिँ इज द भेराइटी टाइप हो त डिफरेंट हुन्छ है त what you need to understand is that binomial nomenclature is not sufficient to name or to characterize a bacteria or a fungi or these microbial organisms so they actually have different further add गरिन्छ यिनीको नाम है त सो यसमा चाहिँ भेराइटी linked भनेर एड गरिएको छ for the solid substitute culture while the Endothea parasitica and the Mucor miehei produce this enzyme in submerged culture. So different methods हरु चाहिँ युज गरिन्छ है त it depends upon industrial है त कुन प्रोसेस बाट जाने भनेर it depends. So renin produced by Endothea parasitica was first renin marketed commercially in 1967. So this enzyme is an acid protease stable at the pH your range 4 to 5.5 with a molecular weight of 34,000 to 37,500 Daltons. Your range ma chai yo enzyme ko chai some certain properties haru cha molecular weight haru pani cha. So what are the different medium kasto huncha? So medium it contains 3% soya milk, right? Protein source soya milk huncha, glucose, right? Skim milk, right? For certain inducers right salts right different salts sodium nitrate K2SPO4 magnesium sulfate different pH incubation temperature it can be generated at the end of 48 hours incubation period mycelium is separated and broth which contain enzyme right is concentrated and precipitated in an evaporation process. Similarly, Mucor miehei also produce renin in medium containing potato starch, soya milk, ground barley, calcium carbonate, and pH 5.5 and 5 to 6 days of incubation period at an incubation temperature of 40. So microbial renins are stable at high temperature and remain active in curd after precipitation and subsequently causing harmful proteolysis. Therefore, complementary cDNA from calf renin is transferred into E. coli, making possible the first commercial production of calf renin by a microorganism. So, since there are some of demerits, like the demerits are currently actually, so you harmful proteolysis gonna sucks. That's why okay. What's the best way? Actually, now we have this technology, genetic recombinant technology. So what we can do is we can sequence, we can clone the information from the calf renin, and then we can transfer it into E. coli, and it's now possible for this commercial exploitation of the calf renin in microorganism. So we have come a long way into this manufacturing process. So biological detergents, it can be used like alkaline proteases, baking industry ma use huna sakcha, right? So dough modification, gluten weakening, and flour improvement, beer brewing, chill proofing of beer to remove the protein haze. Similarly, leather baling and tendering, cheese manufacturing, clotting of milk protein and promoting of ripening, aspartic proteases, chymosin, and aminopeptidases. It's meat tenderization and removal of meat from bones. So meat tenderization ma pani use सक्छ, flavor control and production of food products, waste treatment हरुमा पनि, treatment of silver from the spent photographic film. So film production गर्नेहरुमा पनि these proteases हरु use हुँदो रहेछ है त. These are some of the industrial application of these enzymes, proteases enzymes हरु. So this is the general introduction. General introduction गरेको हामीले यो enzyme को. Now actually यसको चाहिँ process, manufacturing process मा हेर्छौं है त. स्क्रिनमा देखिरहनु भएको छ? स्क्रिनमा देखिरहनु भएको छ?
देखिरहेछ सर.
Okay, अब that was a general introduction है त. त्यो एकदम introduction मात्र थियो. त्यो एकदमै त्यहाँ धेरै इन्फर्मेसनहरु थियो, कम्प्लिकेटेड इन्फर्मेसन्सहरु थियो. एक्चुअली अब फर एक्जाम को लागि चाहिँ एक्जाममा क्वेशन्सहरु छ, इन्डस्ट्रियल प्रोडक्सन अफ प्रोटिएजेस सोधेको छ भने, सो यु डोन्ट नीड टु गो थ्रु अल दैट है त. एकदमै धेरै इन्फर्मेसन थियो है त. अ लट अफ इन्फर्मेसन वाज एकदमै अलिक इन्डस्ट्रियल स्पेसिफिक है त. इन्डस्ट्रीमै गएर काम गर्दाखेरि मात्र युज हुने इन्फर्मेसन थियो, राइट? कति पर्सेन्ट यो पर्सेन्ट, कति पर्सेन्ट त्यो मिडियाको कम्पोजिसनहरु, राइट? इन्क्युबेसन टेम्परेचरहरु धेरै थियो. एक्चुअली यो इन्डस्ट्रियल प्रोडक्सन अफ प्रोटिएजेसहरु दे क्यान बी क्वाइट कम्प्लिकेटेड है त. कम्प्लिकेटेड इन द सेन्स एक्जामको लागि प्रिपेयर गर्दाखेरि चाहिँ क्वाइट कम्प्लिकेटेड हुन सक्छ. जस्ट आई हेभ प्रिपेयर्ड है त. एकदमै एउटा चाहिँ सर्ट एउटा चाहिँ आई हेभ प्रिपेयर्ड है त. So for that, either proteases or Q, what are proteases? These are proteolytic enzymes. So they have different names. Either peptidases or like proteineases or proteases or are defined as proteins with capacity to break peptide bonds. So actually, these proteases or these peptidases, proteineases, proteolytic enzymes, what they do is that they're capable of breaking the bonds, peptide bonds or break on structure. The nomenclature community there is a specific way. The International Union of Biochemistry and Molecular Biology recommends the term peptidase. That is the term. We are using many terms. So we may be using proteolytic enzymes or the peptidases, proteinases, or the proteases, but the UIB, EMB recommends the UB, EMB recommends the term peptidases. The committee also defined proteases as the hydrolases that breaks peptide bond. So actually, proteases or the proteinases, they are hydrolase enzymes, right? And they are classified as class 3. The class 3 ma parcha nomenclature ma. We have already studied the enzyme nomenclature. It's my class 3 ma parcha. And actually, peptidases they fall in the section 3.4 ma parcha. 3.4 ma. Right? Yeah, this is something we have already studied about this in our previous lectures. So, semester we have already studied. So proteases are present in all forms of life, right? Some forms of life, we find these proteases. Even viruses are the proteases enzymes. They don't have the machinery to produce the proteases, but they have that information, encoded information, and then they can use that information they can use that information by employing the host machinery. So that is a very common form with proteases, and they are produced by microorganisms, various plants and animals. Among them, bacterial proteases secure most important place due to the enormous industrial applications. Same sources these microbial sources, especially these proteases, bacterial proteases, they have this important place in industrial manufacturing application. They represent one of the largest group of industrial enzymes and find application in detergent industry, leather, food, pharmaceutical and textile industry, as well as silver recovery and bioremediation process. So actually, these are very, very important enzymes. Right? So now let's begin with the industrial production. Industrial production of the proteases. Proteases are usually produced by submerged fermentation, right? So the this is something we have already studied about submerged fermentation, which accounts for nearly 90% of the production of all the industrial enzymes. The technique involves a substrate in the liquid phase. So we have the substrate in the liquid phase. The solid substrate commonly used are the sucrose, glucose, dextrose, peptones, yeast extract, and the salts dissolved in the water. So these substrate materials, they are dissolved in the water, and the liquid substrate can also be made by molasses, broths, or the agro-industrial waste products such as wheat bran, rice bran, sugarcane bagasse, sugarcane molasses, soybean molasses, residual biomass, and effluents. Right? So this is something I have said repeatedly. The industrial एन्जाइम्सहरू यो चाहिँ बायो ट्रान्सफर्मेसन गर्दाखेरिको एकदमै एउटा सिम्पल पार्ट के हो भन्दाखेरि यु क्यान युज द बायोमास चाहिँ युज गर्न मिल्छ है त यु डोन्ट निड अब इफ यु वान्ट टु प्रोड्युस पेनिसिलिन यु डोन्ट निड फ्यान्सी सबस्ट्रेट मेटेरियल्स अल यु निड इज है त मोलासिसहरू भयो भने इट इज सुटेबल similar case यसमा पनि छ we want the industrial production of the proteases, and what you require are these general agro-industrial waste products. The agro-industrial waste products are these biomasses. They are suitable. So bacteria are suited for submerged production due to the requirement of water for growth. The advantages of submerged fermentation are the ease in controlling the process and the recovery of the extracellular enzyme. Right, so this is the important part. We have already studied about the advantages of submerged fermentation, so this is not something new. Similarly, the enzyme purification is a complex process, and number of methods are usually applied in sequence for the purification of enzyme. To get the pure enzyme after the production of proteases, several purification techniques can be carried out. A wide range of techniques is available for the recovery of the product from the fermented substrate, and the choice depends upon the source of enzyme, that is the intracellular or the extracellular, scale of operation, and the enzyme stability. So actually, from the first step, you can prepare the enzyme. The enzyme can be synthesized. अब यो चाहिँ extracellular हुन सक्छ, intracellular हुन सक्छ, but normally हामी चाहिँ we go with the extracellular enzyme production because that is post recovery चाहिँ एकदमै सजिलो हुन्छ, right? So now let's begin with the concentrating the enzyme. So the primary objective in enzyme downstream processing is the removal of water, because the amount of enzyme present in cell-free filtrate is usually low, so as to the concentration of enzyme becomes high. So first step here, removal of water. Right, so membrane separation process has been widely used for downstream processing and ultrafiltration. The pressure-driven process separation process is inexpensive, results in little loss of enzyme activity, and offers purification and concentration, as well as diafiltration for removal or for exchanging salt composition. So in this way, you can concentrate the enzyme with the different steps, but you can concentrate the enzyme. So one of the first step is the membrane separation, right? Membrane separation चाहिँ एउटा चाहिँ one of the एकदमै easy process हुन्छ, right? So ultrafiltration पनि you can use ultrafiltration, and why we used these membrane separation process, because these are inexpensive. Or also you can use the diafiltration, right? dialysis पनि युज गर्न सकिन्छ. From that you can remove certain salts, right? Salt compositions चाहिँ you can remove from the त्यो broth जुन final broth बनेको छ त्यहाँबाट you can remove. Similarly, the precipitation you have to precipitate that enzyme. So precipitation is the most commonly used method for the isolation and recovery of the protein from the crude biological mixture, right? The crude biological mixture. You have already concentrated it, but it is still you have to precipitate the enzyme, and the precipitation is one of the most commonly used method either. And it also performs both purification and the concentration step. So purification step पनि हो, concentration step पनि हो. So it is generally affected by the addition of the reagents such as salt or an organic solvent. So how is it done? So actually you add the salt or the organic solvent, right? This is the way, and which lowers the solubility of the desired protein in the aqueous solution. So salts like ammonium sulfate and the sodium sulfates are also used, right? हामीले पढिरहेको हो, right? Salting in, salting out को बारेमा भनिरहेको छु the precipitation भन्दाखेरि. I hope you are clear, right? So proteases from the Bacillus licheniformis ATCC 21415, it is यो strain को नाम हो, right? This is the name of the strain. And the Pseudomonas aeruginosa, where manifold purification was obtained from the ammonium sulfate precipitation. So यो दुईटामा चाहिँ के देखिएको छ भन्दाखेरि what we have seen is that यिनीहरुमा चाहिँ यो ammonium sulfate precipitation, it works wonders for these while we are producing proteases from these bacteria. So finally, what we use is the gel filtration chromatography is convenient technique for protease purification from many bacterial strains like Bacillus cereus AT and the Bacillus circulans. So finally, how you can separate? So finally, you can separate with the help of gel filtration technique. You can separate. So this is the figure that you need to make. You just सबैमा एउटै बनाउने हो है त. यो industrial preparation को मा चाहिँ सबैमा एउटै बनाउनु पर्छ. So actually, so the microbial strain हुन सक्छ, submerged fermentation हुन सक्छ, solid state fermentation हुन सक्छ, and the cell separation, biomass and compost हुन सक्छ, and then supernatant, cell-free supernatant मा चाहिँ finally enzyme concentration, enzyme stabilization र enzyme product चाहिँ we can generate. So solid state fermentation मा separation, extraction and separation, and similar हुन्छ. This is how it can work. This is the most common way. So quick questions, quick queries also. Any questions? Any queries? Hello.
Fine, sir.
अब यो industrial preparation भनेर exam मा सोध्छ है त, proteases भनेर they ask you sometimes, कहिलेकाहीँ amylase भनेर सोध्छ, कहिलेकाहीँ proteases भनेर सोध्छ, कहिलेकाहीँ lipases हरु भनेर सोध्छ, र हामी अब series of week हरुमा चाहिँ will study about these है त. यो series यो around 8-9 वटा छ, र हामी चाहिँ will study about these है त. र मैले कतिवटाको चाहिँ I have already added है त, ड्रप बक्समा मैले एड गरिदिसकेको छु. You can go through them है त. यो अब there are these है त. अब आज हामीले amylases we have already studied about amylases, proteases पनि पढ्यो. We study about lipases, त्यसपछि cellulases, त्यसपछि pepin, bromelain, cellulose हरु, different commercial steps हरु, commercial कसरी फंक्सन हुँदो रहेछ भनेर we'll study है त. General principles हरु चाहिँ they are the same. The general principles हरु same हो. You just need to be familiar with the general principles है त. अब actual industrial preparation चाहिँ एकदमै complicated हुन्छ. It's quite complicated, but अब exam मा चाहिँ तपाईहरुको लागि चाहिँ just the general steps हरु के हुन्छ भनेर चाहिँ सोध्छ है त सो this is something you need to prepare है त exam को लागि you need to be quite aware है त so next class मा हामी will study about other दुईटा enzymes हरुको बारेमा पढ्छौं this is will how we will proceed है त केही छ क्वेशन्स भन्नु
छैन सर
ओके ओके देन अब आजको क्लास हामी यति नै गरौँ है त आजको क्लास यति नै गरौँ लेट्स मेट कुनै यो हप्ता तपाईहरुको छ फ्री